It was introduced in Lok Sabha in 2013. The DNA, deoxyribonucleic acid, is like a set of instructions or blueprint of all living forms, and it encodes a detailed set of plans for building different pieces of the cell of a living organism to grow and function. The DNA blueprint varies from one individual to another. It is this variation which makes every individual except identical twins unique and different. The individual to individual variations in DNA permit its use as a means of identification and for establishment of biological relationships between individuals. DNA technology has the potential of wide application in the justice delivery systems. In criminal cases, it helps in investigation of crimes through biological evidence. In civil cases, it helps in investigations relating to identification of victims of disaster like cyclone, air crash. A number of crimes are committed by repeat offenders whose apprehension and conviction will be aided by comparison of biological evidence at the scene of crime with DNA profiles stored in a DNA data bank. At the same time, the DNA analysis offers substantial information which, if misused or improperly used, can cause harm to individuals or society. The DNA Technology Use and Application Regulations Bill 2018 intends to regulate the use of DNA technology for the purpose of establishing the identity of certain categories of persons, including victims, offenders, suspects, under trials, missing persons, and unknown deceased persons, etc. Okay. There is another news that in 2023 or 2024, the bill has been withdrawn from Lok Sabha. Please note. Next, salient features of the bill. The DNA Technology Use and Application Regulation Bill 2018 is divided into 9 chapters, which provides for DNA Regulatory Board, accreditation of DNA lab, obligation of a DNA lab, DNA data bank, etc. The bill also includes one schedule which contains list of matters for DNA testing. Salient features of DNA Technology Use and Application Regulation Bill 2018 are as follows. DNA Regulatory Board. The bill provides for establishment of a DNA Regulatory Board which will supervise the DNA data banks and DNA labs. The Secretary Department of Biotechnology will be ex officio chairperson of the board. The board will comprise an additional 12 members to be appointed by the central government. The DNA Regulatory Board. Next, functions of the board. The functions of the board include 1. Advising governments on all issues related to establishing DNA labs and DNA data banks. 2. Granting accreditation to DNA labs. 3. Supervise DNA labs and DNA data banks including their quality control. 4. Make recommendations to the central government for the application of privacy protection in relation to access to or the use of DNA samples and their analysis. And 5. Give advice on matters under the Act which may be referred to it by central government on the state government or the state government, etc. Next heading, DNA laboratories and its obligations. Any laboratory undertaking DNA testing is required to obtain accreditation from the board. Under the bill, every DNA laboratory is required to perform various functions including 1. Following standards for quality assurance in collecting, storing, testing and analysis of DNA samples. 2. Establish and maintain such documentation and quality systems as specified. 3. Prepare and maintain quality manuals containing such details. And 4. Share DNA data prepared and maintained by it with the National DNA Data Bank and the regional DNA data banks in such manner as may be specified by the regulations. Every DNA lab has to appoint a person to be in charge of the laboratory and employ such a scientific, technical and other staff possessing such qualifications and experience as may be specified by the regulations. Next heading, DNA Data Bank. The bill provides for the establishment of National DNA Data Bank and Regional DNA Data Banks for every state or 2 or more states as it may deem necessary. A regional DNA data bank shall share all DNA data stored and maintained by it with the National DNA Data Bank. The National DNA Data Bank shall receive DNA data from regional DNA data banks and store the DNA profiles received from the DNA labs in such format as may be specified by regulations. The central government shall appoint a director of the National DNA Data Bank on the recommendations of a selection committee to be constituted by the government for the purpose of execution, maintenance, supervision of the National DNA Data Bank. Protection of information. Under the bill, the board is required to ensure that the information relating to DNA profiles, DNA samples, and any records thereof forwarded to or in custody of the National DNA Data Bank or the Regional DNA Data Bank or a DNA laboratory or any other person or authority are secured and kept confidential. The board is required to take all necessary measures to ensure that the information related to DNA samples and DNA profiles are protected against access, use or disclosure or against accidental or intentional destruction or damage. That is about. Next, offences and penalties. The bill specifies penalties for various offences, including for disclosure of DNA information or using DNA sample without authorization. 3. Unlawful access to information in DNA data bank or destruction, alteration, contamination or tampering with biological evidence. For instance, disclosure of DNA information will be punishable with imprisonment of up to 3 years and fine of up to 1 lakh rupees, while destruction, alteration, contamination or tampering with biological evidence will be punishable with imprisonment up to 5 years and fine of 2 lakh rupees. Next, statement of object and reasons. The DNA is like a set of instructions or a blueprint of all living forms, and it encodes a detailed set of plans for building different pieces of the cell of a living organism to grow and function. The DNA content of every human individual is comprised of one half of DNA from each of the two parents. The DNA blueprint varies from one individual to another, and it is this variation which makes every individual except identical twins unique and different. The individual to individual variations in DNA permit its use as a means of identification and for establishment of biological relationships between individuals. 2. DNA technology, based on sound scientific principles, has been found to be very effective in establishing the parentage of a child and identifying the source of a biological specimen obtained from a scene of crime. The concerns regarding appropriate use of DNA technology by the courts of law and other agencies has made it necessary to develop guidelines and standards for the DNA testing. 3. DNA technology has the potential of wide application in the justice delivery system. In criminal cases, it helps in investigation of crimes through biological evidence including semen evidence in rape cases, blood evidence in murder cases, saliva evidence in identification of source of anonymous threat letters. In civil cases, it helps in investigation relating to identification of victims of disaster like cyclone, air crash. A number of crimes are committed by repeat offenders whose apprehension and conviction will be aided by comparison of biological evidence at the scene of crime with the DNA profiles stored in a DNA data bank. At the same time, the DNA analysis offers substantial information which, if misused or improperly used, can cause harm to individuals or society. Recognizing the need for regulation of the use and application of DNA technology, a DNA profiling advisory committee comprising of members from fields of molecular biology, forensic science, human genetics, population biology, bioethics, legal profession, law enforcement agencies was constituted in December 2003 to make recommendation for enacting a suitable legislation. On the recommendations of the said committee, a draft bill was prepared. Later on, an expert committee was chaired by the Secretary, Department of Biotechnology, was constituted in 2012 to discuss the privacy related issues. Based on the recommendations of the expert committee, the bill was revised and subsequently referred to the Law Commission of India, who in its 271st report suggested the enactment of a legislation. In a view of the above, the DNA Technology Use Application Regulation Bill 2018 seeks to regulate the use of DNA technology for the purposes of establishing identity of certain categories of persons, including the victims, offenders, suspects, individuals under trials, missing persons and unknown deceased persons. The bill inter alia seeks to 1. Prohibit laboratories from undertaking DNA testing, analyzing without obtaining accreditation. 2. Establish a national DNA data bank and regional DNA data banks which shall store and maintain the DNA profile in accordance with the provisions relating to use and access to information, retention and expulsion. 3. Establish a DNA regulatory board to carry out the functions assigned to it under the proposed legislation, which inter alia will include A. Advising the central government and the state governments on all issues relating to establishing of DNA labs and DNA data banks, and laying down guidelines, standards and procedures for establishment and functioning of such labs and data banks. B. Granting accreditation to labs for undertaking DNA testing, analyzing, and to suspend or revoke such accreditation. C. Assisting in criminal investigation between various investigation agencies within the country and with any foreign state, international organization or institution. And D. Making recommendations to the central government for the application of privacy protection in relation to the access to or the use of DNA samples and their analysis. 4. Make provision for the security and confidentiality of information relating to DNA profiling, DNA samples and any records thereof forwarded to or in the custody of National DNA Data Bank, regional DNA data banks, DNA labs or any person or authority. Provide for offences and penalties for contravention of certain provisions of the bill. Here are the above. This is the bill and objects. Privacy activists fear it will lead to gross violation of human rights. And the history is 1985. Indian courts accept DNA as evidence in criminal investigation. 2003. Work begins to draft a bill to regulate use of DNA samples in probes. 2005. Code of Criminal Procedure amended includes use of DNA profiling. Government says bill will be useful in 1. Crime scene investigation, 2. Maintaining database of convicts and suspects, verification of missing persons and investigation of unidentified bodies and research work. Activists claim that the bill could lead to 1. Racial and communal profiling, violation of privacy and longer trial periods and errant testing and conviction. Central idea. The Union Government recently withdrew the DNA Technology Use Application Regulation Bill 2019 from the Lok Sabha for revision. DNA Bill 2019 highlights the bill first proposed in 2003 aims to establish a regulatory framework for obtaining, storing and testing DNA samples of individuals primarily for criminal investigation and establishing identity. Over the years, the bill has undergone changes and was referred to Parliamentary Standing Committee in 2019. The committee raised concerns about potential misuse based on religion, caste or political views. Key features. Objective. The bill sought to create a regulatory framework for DNA sample collection, testing and storage, primarily for criminal investigation and establishing a person's identity. 2. Existing uses of DNA technology. DNA testing is already employed for criminal investigation, parentage establishment and locating missing individuals. 3. Proposed institutional structures. The bill aimed to establish a DNA regulatory board and a DNA data bank at a national and state level. Role of DNA regulatory board. The board would frame guidelines and rules for DNA collection, testing and storage. Next, DNA data bank. The data bank would store all DNA samples collected under specified rules. Restricted testing. DNA sample testing would be allowed only at laboratories authorized by the regulatory board. Others will not be allowed. Next, handling of DNA samples. The bill specified the circumstances under which individuals could be asked to submit DNA samples, the purpose of such requests and the exact procedures for handling, storing and accessing these samples. Next heading. Controversies and objections against the bill. Reliability of DNA technology. Critics raised concerns about the foolproof nature of DNA technology and its potential for error. Risk of misuse. The main debate centered on the possibility of abuse of DNA information. Detractors feared that the intrusive DNA data collection and storage could lead to misuse and violation of individual privacy. Privacy concerns. DNA information reveals not only a person's identity but also physical and biological attributes like eye, hair, skin color, susceptibility to diseases and possible medical history. Critics argued that storing such personal information could compromise privacy rights. Next heading. Standing Committee's concerns. 1. Technical and sensitive nature. The Standing Committee's report acknowledged that the bill was technical, complex and sensitive. Addressing fears, the report recognized and addressed concerns expressed by several members about the potential misuse of DNA technology based on factors like religion, caste or political views. Next heading. Government's defense of the bill. International precedents. The government argued that nearly 50 countries have enacted similar legislation justifying the need for such a law in India. Limited information storage. The government contended that only a limited set of numbers, just 17 out of billions that DNA samples can reveal, would be stored on the indices. This information could act as a unique identifier, not reveal any personal data that affect the privacy. Conclusion. The withdrawal of the bill marks a pause in the government's effort to create a regulatory framework for DNA technology regime. The controversies and objections raised highlight the need for a balanced approach. Centre must address concerns over misuse and privacy while harnessing the potential benefits of DNA tech for criminal investigation. Now back to basics. DNA. What is DNA? DNA, deoxyribonucleic acid, is a molecule that carries the genetic instructions necessary for the growth, development, functioning, and reproduction of all known living organisms and many viruses. It is referred to as the building block of life. Key features of DNA include molecular structure. DNA is a double-stranded helix molecule consisting of two long chains of nucleotides that form a double helix. Each nucleotide consists of a sugar molecule, that is deoxyribose, a phosphate group, and one of the four nitrogenous bases, which are adenine A, thymine T, cytosine C, and guanine G. Base pairing. The two DNA strands are held together by hydrogen bonds between complementary base pairs. Adenine A always pairs with thymine and cytosine C always pairs with guanine, that is ATCG. Generic code. The sequence of nucleotide bases along the DNA strand constitutes the genetic code, which determines the specific traits and characteristics of an organism. Genes. DNA is organized into specific segments called genes, which are responsible for encoding proteins or functional RNA molecules. Proteins play a crucial role in various biological processes, while RNA molecules contribute to gene expression and protein synthesis. Replication. RNA has the unique ability to replicate itself through a process called DNA replication. During cell division, the DNA unwinds and each strand serves as a template for the synthesis of a new complementary strand, resulting in two identical DNA molecules. Inheritance. DNA is passed from one generation to the next through the reproduction, ensuring the transmission of genetic information from parent to offspring. Role in protein synthesis. DNA provides the instructions for protein synthesis through a two-step process. First, the information in a gene is transcribed into a messenger RNA molecule. Then the mRNA is translated by the ribosomes in the cell to produce specific protein as ordered. Next, genetic variation. Mutations or changes in DNA sequence can lead to genetic variation within a species. These variations are essential for evolution and adaptation to changing environment.